Hello, everyone, and welcome to BrainMap. As you all know, BrainMap is co-sponsored by the P41 Funded Center for Mesoscale Mapping housed in the Martinos Imaging Center. With that, it is my great pleasure to introduce our speaker today, Professor Zhongming Liu. Zhongming Liu received his BS and MS in Electrical Engineering from Zhejiang University and his PhD in Biomedical Engineering from the University of Minnesota. He was then a research fellow in the Advanced MRI section at the National Institute of Health. From 2013 to 2019, he was an assistant and then an associate professor in both biomedical engineering and electrical and computer engineering at Purdue University. Since 2020, he has been an associate professor in the Department of Biomedical Engineering, the Department of Electrical Engineering and Computer Science at the University of Michigan. His lab develops and uses advanced techniques for imaging, recording, stimulating, and modeling the brain to accelerate progress in neurosciences, neural engineering, and artificial intelligence. I would like to remind you all to please answer your questions at the Q&A or just raise your hand and we'll get to you. Without any further ado, I will hand it over to you, Dr. Liu, and thank you so much for being with us today.
All right, thank you for the invitation. It's my great pleasure to share my research with this group. And I had friends in Martinos centers, and I know of the many awesome work that this center has generated and contributed to the scientific community, especially in the field of neuroimaging. So today I'd like to share some work that my lab has been doing in the past several years. And using fMRI to image the brain, to understand the brain, and in the same time try to make machine smarter. All right, so we get started. So the focus of our research is to hopefully generate a two-way facilitative interaction between AI and neuroscience. And I'll touch on three aspects. One is the neural models and processes for vision and language, in the same time, models for understanding brain imaging data. So my interest in artificial intelligence started from 2014. And back then I had a colleague, Eugenio, share with me some of his work that using a handheld smartphone and moving the phone around in the same time recognize all the objects was seen in front of the camera. And also in the real time, and the camera connected to the phone can actually segment objects and cars and pedestrians and streets, for everything in real time. This was quite amazing to me back then. And there has been tremendous progress ever since, and the progress has, changed the world, and it's generated big momentum in almost every single aspect of life and work. So under the hood, at least back then, that the models that drive all this advancement in computer vision was the convolutional neural nets. One of the general strategy is that given an image or a video frame, the pixel patterns enter to a neural net and one layer at a time in a feed-forward direction, and runs through convolution and some nonlinear activation functions, so on and so forth, and generate a representation that eventually lead to recognition of the image. And in many ways, that this model and seems to detect some interesting features. The features goes from very simple features like edge detection and of different orientations and different color, and into something that is a little bit more complex, like the contour and shape, and progressively into something that is more indicative of the object in the picture. And so from the lower layer to the higher, deeper layer, and the information being encoded appears to be increasingly abstract. In many ways, this seems to echo what the ventral stream of the visual cortex does, and across different visual areas and information, visual information processing proceeds towards perceptual understanding of everything we see. So there had been a large number of studies since 2014 in trying to investigate the correspondence or mismatch between the neural network model and the ventral stream of the visual cortex. And my lab had been interested in this field as well. And so the specific question is that how similar those neural nets are to the feed-forward neural processing along the ventral visual stream. So as a researcher that has been used to using fMRI to study the brain, and we scanned human subjects watching naturalistic videos. So we built back then a fairly large data sets that for each individual that we have presented over 9,000 different video clips that we handpicked from the YouTube. And we assemble those video clips into different sessions, and each session is about 8 minutes long. And then we generate large data sets of videos. And it's about like 13 hours of videos. And we present those videos to a human subject. And we also prepared about 40 minutes of videos that we use for testing how the model works. And so it was back then when we complete the data in 2016, this was back then a large data set for natural vision. And the questions that we ask is that when we present those videos to the humans, and we scan the human brain across the entire brain, and we focus on the visual cortex, and we ask the questions that how much the brain responses can be related to the model's responses. The models are convolutional neural nets, and the model watch those videos frame by frame and generate their internal representation and dynamics. So we use those model dynamics trying to predict how the brain will respond to those videos, and we call that encoding. The approach we do is rather simple. So given a video frame to the model generates a pattern of activation, and we use those pattern of activation inside a model and linearly combine those artificial neurons and to predict each individual voxels in the human brain. And this type of regression model is, must be very familiar to many of you guys, that is similar to general linear models, except that the weights of those linear models are sparse given a L1 normalized regression model estimation process. And then we train the model, and then we test how the model, given the neural network and given the stimuli, and can predict how the brain would respond to the stimuli. We use a held-out testing movie to test the model's performance. And to my surprise, and back then to all our surprise, that the model can really predict the voxel responses very well. And you pick up a voxel from V2, V3, V4, fusiform phase area, and among other areas in the brain, in the visual cortex, even though those areas has very different time course, and the model can predict it very well, even though the model has never seen this movie before at the training time. And then this type of model can be really useful. We know that there are many, artificial and carefully designed stimuli being useful to map the retinotopic relationships on the visual cortex. And in fact, that even with natural stimuli, given this type of model, we can easily generate maps of polar angle representation eccentricity representation that seems to align pretty well with classical ways of mapping those retitonopy. And beyond that, you could also look at how different layers are predictive of different voxels. For example, in the early visual areas, V1, and the voxel in V1 is most predicted by the layer 1 in the model, and some of the other area, like say this voxel in V4 is most predicted by a middle layer, and some other voxels that along the ventral stream and can be most predicted by the deeper layers. So you can actually use this type of models to go beyond the conventional way of mapping the visual field. And now you can actually start to look at the hierarchical organization of the visual cortex. Now, in the model, in the convolutional neural nets, and we identify a neuron that tend to respond to the face. So for example, there's a face neuron that whenever in the video, there is a face showing up, and that neuron will be activated. The time that neuron, the activation of that artificial neuron in a convolutional neural net seems to match very well with the fMRI responses from a voxel within the fusiform phase areas. So you can compare this, brain responses on top and the artificial neurons response in the bottom, and you can see that they are fairly close. Now, this type of similarity is not just limited to fusiform phase area. If you map out all the similarities across the brain, you'll find out a network, and that network actually matches very well to the network that will respond to the human face through many classical, face selective stimuli and the functional mapping experiments. And you can actually preserve some of the asymmetry. For example, in this case, that the right hemisphere has more activations to the face than the left hemisphere. And all of this is by simply finding out a neuron in the artificial neural nets, and that neuron actually respond to face and correlate that neuron's responses to the fMRI activity, and you generate this type of patterns. So I think this is a nice intermediate step between the classical ways of using handcrafted or manual features from in a general linear model analysis, and to a more of machine learned, learnable models. And now you can actually take use a similar approach, but ask a question in the reverse direction. And that is, if you have the brain scans, can we actually reconstruct what the human subject was seeing, or extrapolate that concept to other domains, what the human subject were thinking or experiencing? So the approach is very similar. So we have the brain scans, we combine those individual voxels responses, and through a linear model, and we add the sparsity constraints to those linear models. We train them with the video, with training data, and then we test them using the held out testing movie data. And it turns out that this approach can be used to reconstruct certain patterns. It's not really that great, but those patterns seems to match what certain features of what the human subject was seeing. So you can see that those color pictures, what was the video frames that the subject were really watching, and the model reconstruction can show, and there's no color information there, but it can show some, the basic shape and of the objects in the video frames. Now you can predict the features in a deeper layer, and through those layers, you can actually output the semantic information regarding what the human subject was watching back then. So for example, here shows the top 5 concepts that describe what the subject was watching. And showing in red is what the ground truth. In this case, that the model predicted that the subject was looking at something like a bird as the most likely concept, and a visual percept, and then some water animal, airplane, land animal, or something indoor, right? So in general, you can see that the top 1 output seems to match the actual images or the video frames, and so provides a semantic description of what the human subject was watching at that time. Putting all of these things together, so you can actually see, retrospectively and frame by frame, every TR, decoding what human subject was watching as a pixel pattern, which doesn't look so great, but you can see something roughly consistent, as well as semantic concepts, and showing on the right, and doing so in pseudo real time, and that's actually plausible. Now, following those studies, and we actually develop other models called using the variational autoencoder for decoding through an encoder and decoder architecture. And without going much of the details, and we have made some improvement that you can start to see more details, start to see the color coming back, and but still not so great. And we'll guess, but then back in the recent couple of years, there has been lots of other related work, and some of them using the data set that we have generated and publicized, and made better, image reconstruction or video reconstruction. So there's tremendous progress, encouraging progress in this direction. We realize this is a nice tool for mapping brain function in relation to dynamic natural vision. However, that we quickly realized those models' limitation. And those models are, far like the human brain. There are many things that those models differs from the human visual cortex. And one way it differs, I like to talk about this using a simple experiment. Let me try to see if I can hide this information. All right. So for example, you see a picture on the right, and I'm not sure how many of you have already seen a picture like this, but when I first saw this picture, and I had a hard time recognizing what this picture is about. But then if I tell you that this picture has a dog, then you can quickly spot where the dog is, right? Given the dog, you can make further inference that the dog probably is sniffing on the ground, and there might be a tree somewhere in this area of the picture. So the concept of a dog is really effective in guiding the recognition of what this picture and understanding the whole scene. And this is because that the brain, unlike a feed-forward neural nets, and it's not feed-forward only, you have information that enters from your retina all the way to the higher areas in the ventral stream, and that actually will lead to a visual percept and a semantic understanding of the picture. But in the meantime, that those higher level representations will pass information to the lower layer through feedback connections. And this process is dynamic, so it goes on and on. The feed-forward and the feedback, they interact through the recurrent computation. And this type of computation and has the power or the potential to explain many of the phenomena that was regarding the visual perception. One of the phenomena is bistable or multi-stable perception. Give you this example. So if you see this picture, and this picture has two different interpretations. One interpretation is that this picture describes a rabbit, right? You can see this part of the picture, you can see this is a rabbit. Or sometimes you think this picture describes a duck. And if you focus on this part, and both visual percepts are quite likely. You have face and vase, and you have lots of such kind of examples. One theory that has been influential and preferred by many neuroscientists in vision field is what is called predictive coding. And it's a process that describes the feed-forward and the feedback processes using a very specific ways. That the feed-forward feedback process predict the lower layer representation through the higher layer representation. And the higher layer representation calculate, or the lower layer calculate the error of the prediction and send the error prediction forward to further refine and update the higher layer representation. And this process goes on and on and on. Coming back to this example with bistable perception. So predictive coding might be a way to explain this. Let's say that for whatever reasons that someone's visual systems is initially biased towards a rabbit given this picture, that the percept described this as a concept of rabbit, the rabbit trying to explain away the visual input. And then, this part of the picture, this is more characteristic of a rabbit, and this part is a little bit less. So maybe this part of the pixels are left out and then become the errors, and the errors of predictions are more indicative of a visual percept that is a dog. And then this actually eventually will flip the visual percept from rabbit to dog, and this one, and then try to use the concept of dog to explain away the image. And then that will lead to error prediction being more indicative of rabbits. And so that's how probably that the visual percept alternate between two different possibilities, rabbit versus duck. So the top-down prediction and the bottom-up recognition always trying to reconcile. The way they reconcile is through the computational principles for inference based on predictive coding. And so that's one, at least that what I prefer thinking of. There has been a lot of studies from theoretical neuroscience and neurophysiology, and as well as neuroimaging, especially layer MRI, fMRI, and trying to relate this feedforward feedback computation or predictive coding to the canonical circuits within a cortical column across different layers. And we were inspired by this type of layer-specific computations, so that their different layers are involved in different ways in the feedforward and the feedback calculations. And we designed a model that actually tried to do the same. So in the model that we have 3 types of neurons. One type of neurons receive predictions. The other type of neurons we call state neurons that hold up the representation. And then the difference between these 2 groups of neurons is calculated in terms of the error of the prediction. And the error of prediction is sent in the feedforward direction to the next layer. In the visual cortex, visual areas, it's more of similar to a layer in a neural network. So we developed this type of models, and we developed a way of a dynamic recurrent processes for running inference based on the principle of predictive coding. And this process can unfold itself over time, so generate dynamic visual percepts. And we train this model to perform visual recognition with CFAR 100, and then followed by ImageNet 1000. And we train those models, and we find those models actually generally perform better than the convolutional neural nets. And the convolutional neural nets is actually the backbone of the model that when we and we extend and refine it as a bidirectional feedforward and feedback network. It performs better, and the performance tend to improve given longer time for visual inference. To give you a more concrete example, so when you see a picture like what is shown here, that the visual percept at the first time, it's not as decisive. And as the time goes, the visual percept becomes more and more decisive and settle on a visual percept that is, this picture is about a car. Similarly, this picture, I mean, when I look at this, I could not even tell whether it's a cat or a dog. And this seems to confuse the model too. But eventually the model settle a visual percept that this is a dog. So I believe this, is a little bit progressive refinement of the visual perceptual decision over time. Seems to be relevant to how we recognize our visual surroundings. And at least back then, that there was a trend, still the trend remains that the model's getting deeper and deeper and wider. But generally, the model remains unidirectional, that goes from the input all the way through the output. And I personally believe, and a more of a brain-like model will be bidirectional. So there are information processing and through input to the output, but there will be output back to the input, and then close the loop and do so recurrently. So almost like a fixed hardware or architecture is reused, revisited recurrently, such that this static architecture can unfold itself over time to give you a deeper and deeper neural network. So we have been keep pushing this direction with more ongoing work, which I hope will present sometime later. And that's not the only differences between the brain and the model. And there are many other differences. And there's another difference that I think is very worth noting, is that when we see a visual surrounding, we are not always, we don't define the center of the picture as where we look. Unlike a camera and computer vision models, they always see and take in all the pixels all at once. Instead, humans is going to look around. For example, this one, sometimes you look at the duck, sometimes you look at this frisbee, and as you look around, your eyes move around, and the projection of the visual surrounding onto your retina will look more or less like this. Okay, so humans and most of the animals see the visual world through some sort of fish eye, and that has never or has rarely been taken into account in most of the models in computer vision. And so, as someone who's not just interested in computer vision or AI, I'm interested in understanding how the brain performs those tasks. So we noted that, the eyes is a big difference on the front end between the brain and those models. But the retina has different retinal ganglion cells, and they have different spatial and temporal sensitivities. And those cells actually pass information to different layers in LGN, which is the visual thalamus, and those information remains segregated and further pass onto the primary visual cortex. And then after the early visual areas and those information start to diverge, and one move along the ventral stream, the other move along the dorsal stream, and information passing along the dorsal stream would actually control the eyes so that, and control the eye movements through the oculomotor system, so that we're going to start to move around on a scene. So we inspired by this, neuroscience knowledge or evidence, that we built a model that does not include only one stream, but include two streams. One stream, where we call it the where stream, where CNNs, is take a broad view and then knowing where to look, where in the scene that are important, and then move the eyes or move the center of fixation to the next position that remains salient. And then as the eyes moves, and the other stream, which we call the what CNS, it's gonna take a refined sample, but let's focus more on a local view and amplify the visual information around the fixation and process this information. And then the two streams interact in such a way that the ventral stream will accumulate information across multiple fixations and to generate an understanding of the objects or the scene. And so we built this model, and the model actually can do a good job figuring out where to look next, So figuring out where are important locations to look. So for example, these are a video, Indiana Jones, and you can see that the model will recognize the important parts in every video frames, and those important parts tend to be locations that we're going to look at. And the model can actually generate the fixation patterns similar to the human's saccadic movement. So actually, for example, the model will first look at this part, and then look at this part, and look at that part. And for this sleeping cat, and you have different eye movements, that seems to appear to be very reasonable, at least to me, and how I would look at different parts of an image. And with that, we train this model. We realize, interestingly, that the where CNN, which is one string in the model, that tend to predict better certain voxels on the cortex, where the what CNN, which is the other string in the model, tend to predict better another set of voxels in the visual cortex. So putting together, these are all the voxels that the model can predict. And those voxels are all the vision-related cortical locations. But then the ones that the where CNN can better predict, it tends to align themselves along the dorsal pathways, where the ventral stream would tend to better predict the voxels along the ventral pathways. So this was encouraging because it's a new finding that we find how a model that's trained to do certain things like the human's visual behavior, and will actually be able to explain the segregated functions of the visual locations, visual cortical locations along those dorsal and the ventral cortical pathways. And then we ask why that happens. So this is another summary of this, and different visual areas along ventral and dorsal pathways are better predicted by one or the other visual streams in the model. We asked why. So we did lots of ablation studies. And the primary reason is the different functional roles, the two CNNs in the model and trying to perform. One is to locate the objects, where to look next, which is the spatial attention. And the other is to recognize what the object is, which is the visual perception. And the difference in the learning objective is the primary driving factor for the functional segregation of the dorsal and the ventral streams, as explained by this model prediction, in line with many years of the visual neuroscience studies. Now, to turn the gear from vision to language, and language is how unique to humans, and we communicate by language. We read and we speak language. So language understanding has a lot of times something fundamental to the human cognition. So after we do these dynamic natural vision experiments, we did another set of experiments where we scan the human brains, and we while they were listening to an audios, and we tried to take a similar strategy and trying to bridge the model for natural language comprehension to the cortical responses in those humans while they listen and comprehend those audio stories. A little short, and we have about 80 different stories from a moose radio hour, which is an natural story, audio story data sets. And we scan 19 healthy subjects using 3T fMRI. And then we actually look at the cortical responses, and we look at the representations of individual words in the natural speech stimuli. And we're trying to build a linear regression models and to connect them together. I mean, similar to how we have done using for natural movies. And then again, and this time that the model seems to perform really well. And for certain cortical locations, the locations that can be predicted by the model tend to align pretty well with what we know as the semantic system on the cortex. And those systems are highlighted here. And then not only that it aligns well with the semantic systems, but because the models encode the different type of words, we can use the model to map out how the semantic systems represent different type of words or concepts. And we realize that the left hemisphere tend to better encode concepts that are more concrete, like the tools, animals, plants, and humans, while the right hemisphere tend to encode concepts that are more abstract, like quantity, change, emotion, communication, so on and so forth. And then we could also map out what is the cortical process that the brain may perform or engage in certain cognitive inference. For example, how when the brain is trying to do an inference from parts to whole, examples like finger, it's a part of hand, an animal, it's a part of zoo, and second, it's a part of an hour. This type of conceptual inference, seems to be associated with the deactivation of the frontal parietal network and the default mode network. And this was actually, I think that some of the other benefits of using this type of modeling strategies for understanding for interpretation of the human fMRI signals in terms of cognitive inference. And we didn't go too far in that direction because every time when we look at a model, we quickly realize where the model falls short in terms of capturing how humans represent information, understands information. For example, and when we learn the words, and at least when I learned the words, and I often was told that this is a word banana, I was often shown a picture, this is a banana. So a lot of times that we learn concepts, not just by reading, by listening, but at the same time relating those concepts to what those concepts imply in the physical world, in terms of how it look like, how it feel like when we touch it, how it smell like, So we, all these physical properties and attributes are associated with individual concepts. And those concepts are not simply symbols, but a collection of attributes that are grounded to the physical world. And there's rich literature and in semantic grounding, and that's inspired us to build a different model. And we published that model in 2021. A model actually trying to bridge the vision and language models through contrastive learning and the cross-model learning. And around in the same year, and OpenAI actually, published also a model as CLIP that's of bigger size and actually with similar concepts. We do this model. Our model was performing this type of learning through multiple stages, but essentially is trying to find out how, given an image and a sentence that describe that image, how the model could actually associate the sentence, each word in the sentence, to every location on the picture, right? And that association need to be dependent on the context. Give you one example. So this is a picture, a cat standing on a bathroom sink, and a sentence that describe it after learning is a black and a white cat standing in a white bathroom sink. And the individual words are associated and grounded to different parts of the picture. For example, black here shows a black, and the white here shows a white, and a cat here shows a cat, and you have another white. The second white, these two white are the same word, but the second time the white, is showing, the edge of the sink. So now the same word, same concept, given their different contexts, are associated and interpreted as with different visual correspondence, right? One refers to a cat and the white fur of the cat. The other refers to the white color of the sink. So this shows some of the intrinsic attributes of this type of model. And the model is also able to make inference about the relationship between different objects in the image. For example, the donut is on the table. The model figured that out, and the donut is maybe next to a cup or something like that. And this type of relationship, the model will be able to make inference about. Now, once the model is trained in such way, the model can do more things. One thing that we entertain ourselves, we test it, is that, if you see a picture like what is shown here, with some, black and white stripes and a word, horse, and what that combination would imply, well, we could actually tell that this implies something maybe like a zebra, right? If we consider this picture 50%, this horse 50%, and that will give us a clue that this is probably a zebra. The model, by grounding the vision, language to vision, is able to represent the visual information and semantic information in a joint representational space. So that, if a horse is a vector in that space, and this picture is a vector in that space, you can actually combine these two vectors in terms of their weighted sum. While the weight, you can go from being heavy weighting for the picture to the heavy weighting for the word. And as you do that, you find out the pictures that matches these representations. You find if this, is a heavy weighting with the picture, you can see a lot of striping things, but not necessarily always the zebra. Now, you end up seeing something like a zebra when you have equal weighting for the word and the picture query. But if you have heavy weighting for the word without the consideration of the picture, you end up being only the horse. Now, we can do this for all kinds of word and picture combinations. For example, a picture of cat, a word with sleeping, you can change the weighting from picture, heavy weighting on the picture, towards heavy weighting on the word. You can end up being the cat goes all the way into a sleepy cat, into something that is sleepy dog, and you have a coffee and a milk. You end up being cappuccino, something latte, and in the middle, and you have a black coffee on the left. You have a pure milk on the right. A car and a Lego, you have Lego car in the middle. And so you see this continuous variation of the concept, and you can generate that. And we didn't explore that, but a lot of people following the similar ideas using clip models, being able to generate all kinds of pictures given the text, prompt, things like that. And I know that I believe that many of you guys probably have explored some of those tools, like the ChatGPT. Now, being someone interested in neuroscience and looking into, after you have the semantic space grounded to vision, does that inform us anything about how the semantic space is structured? So we did a PCA analysis, and we find that semantic space has multiple principal components. The first principal components seems to align very well along concepts that goes from abstract concepts to concrete concept. So you see that cooking tools, horse and house, and then you see the emotion, big, small, mass, and those are very abstract concepts. And you can do this for the second principal components, which seems to indicate human versus non-human objects concepts. The third principal component seems to indicate object versus scenes, or animate versus inanimate concepts. And then you can actually use those concepts, mapping them all back to the brain using the fMRI data that we have collected, the natural speech stimuli. You can actually figure out something about how different type of concepts, different dimensions of concepts are encoded by in a distributed manner by the semantic system. So we believe this could actually have some values in cognitive science. Now, the last piece, and I'm not quite sure that how I'm doing in time, but in the end of it, I like to touch on the last piece of how we develop models to analyze the fMRI data and trying to understand how the brain networks and the functions of those networks and the network dynamics. So we look into a model which we originally developed for natural vision and connected to the fMRI data during the naturalistic stimuli. But this time we built a variational autoencoder to encode and represent the fMRI activity patterns. And going to little details, that if we have the cortical representation of a cortical activity pattern, we inflate it into a sphere, like what free surfer tend to do. And then we actually unfold this sphere based on, the two angles. One is like latitude, the other is longitude, and we actually represent this the spherical representation as a two-dimensional grid, right? Like how we draw the map of the globe on a piece of paper. And then we use a fairly straightforward variational autoencoder. We implemented that, and considering both the left and the right hemisphere, and we learn representation in the middle. And this representation gives us an alternative way of looking at the cortical fMRI patterns, not by voxels, not by ROIs, not even by resting state networks, but by a vector that is 256-dimensional vector, much lower dimension than the original fMRI pattern, and to trace how that vector evolves over time as the activity patterns go, up and down, evolving in time during an fMRI scan. And this one shows the resting state fMRI data and from the Human Connectome project. And as we play this out and then project the data and then present the data in this latent space, we realize, interestingly, that even though the model training is look at the data from 500 subjects, it turns out that a lot of subjects are readily differentiable on this latent space. So here shows 20 subjects, and each subject is color-coded as a bunch of patterns in the latent space. We could almost see that these patterns are differentiable by color, which means subject, in terms of where they are, in terms of the shape of representation. And then taking that as a clue, that we were able to use the latent representation to identify those individual subjects, and in a way that is better than identifying those subjects using what is called the connectome-based predictive modeling, CPM. And this performance, performance difference is particularly large if we're dealing with, subject identification for a lot of subjects, like 500 subjects or something. And we don't necessarily need to use a lot of data. We need only use about, 3 minutes of data. We were able to achieve 90% of accuracy in individual identification out of 500 subjects. So I think this is an advantage for understanding individual variation, even though the model is not explicitly trained to do so. Now, we also find that this actually captured a lot of brain dynamics. So these are changes of the brain states from, looking at a blank screen to start looking at a movie, like, from, for 30 minutes, 30 seconds of data. So then across about 173 subjects, we can see a yin and a yang type of transition of the latent trajectories goes from one side of the latent space to all the way to another side of the latent space. And that type of brain state transition dominate the individual variation. So we can start to look at this with many different stimuli, and HCP data sets has many movie stimuli that we can play with. And we can see that how the latent representation jump from one to another location, and those jumps seem to correspond pretty well to the transition of the content in the video frames. And we can do this for different type of movies. These are all the HCP data sets. You can see that the jump tend to be very dramatic when in this Inception movie, that there's something very sensational, non-realistic happening in the movie. And certain type of scene does not have a lot of maybe arousing content, and you don't see as much of the transition. We use this type of transition, which we quantify as a gradient, to characterize the dynamics of brain activity. We find out that we can almost rank those individual movies based on how arousing, how much of the dynamics that cause in the brain. And that seems to be fairly robust and reliable, and seems likely informative as well. So we believe that this is a beginning of having a tool that we hope we could continue to develop. And we have lots of ongoing work, and some of them have been published. I'll leave it out for this presentation. I'll stop here. And to cap that all this, some of the examples of our research in this direction, which our first paper was in this direction, was out in 2016, and it has been found several years, and we believe we will continue and explore at this intersective areas between neuroscience and AI. And with that, I would like to say that my lab is now moving into different modalities, different cognitive systems, and using the electrophysiology, fMI, especially layer fMI, and trying to look at further details in the brain through their relationship with models of neuroinformation processing. With that, thanks to all the students currently in the lab and those who have already moved on to their own career. Some are founding, and thanks for your attention, and thanks for having me again.